Thank you very much, Marty. I always enjoy these and learn a lot. So my name is Thomas Farb, and I'm here to talk to you about topics very similar to what was just discussed, which is, however, in a field that has been neglected, which is live cell biology, cell culture, live cell imaging. We started Thrive because we found this critically important area, which was not digitized, and it's not often that you can find something like that. Just a little bit on my own background, I've started 17 companies. I've had 7 unicorns. Of those 17, I've been on the investing side. I ran a fund with a sole LP, the city of Shanghai. But what I really like to do is, start companies and also angel invest. I was the first angel investor in a company called Wuxi AppTech when it was about 10 million in revenue instead of the billions now. So I like building value for the long term, and we found this area which is really neglected, and the importance of it is that cell culture and cell culture imaging is really key to biomedical research. Yet it is conducted manually, like it was 70 years ago. And we take these cells that have variable quality and poor quality, and we put them into very expensive instruments. We spend billions of dollars studying cells that are really defective, really poor quality. So we have the status of preclinical research is that we have incredible variability of results across researchers, across labs, across time. We have extraordinary contamination of cell lines. A study that was done by the FDA found that HeLa DNA was in 90% of cell lines. We have a crisis with mislabeling. So another study by NIH found that 11% to 30% of all cell lines were mislabeled. So what does that mean? That means that you think you're studying and doing drug screening on liver cells, but in fact, they're pancreatic cells. And so this explains a lot of why there is such low odds of success in drug discovery. A lot of these things are only caught once we're in the clinic, which is extremely expensive. So let me tell you just a little bit about how a cell culture lab works. Cells are kept in these incubators that are not smart. There's no intelligence built in, they're not networked, and the cells are taken out and looked at under a microscope for a couple hours a day by researchers. And they cross multiple environments, which creates genomic drift and stress. And then they're put back after a couple of hours of being looked at, maybe a couple times a day. And as soon as they change temperature, there are shock proteins that are being emitted. They're put back into this incubator. And guess what? Virtually no data has been kept. Maybe some scribbling in lab notebooks, maybe occasional digital microscopy shots, pictures, but that's not full data sets. And what we're doing is creating for the first time full massive data sets easily in live cell biology. I would say that Uber knows much more about us than we know about the cells that we're studying, and those are cells that we're studying to bring medicines to people. So lucky I have my slides here that I can look at.
You need me to put them on your share screen?
Yeah.
Okay, just give me a second.
Okay. Sorry, I didn't realize I needed to tell you that. So what is the result of this terrible environment for live cells that I just talked about, this manual environment with mislabeling and stress? Well, the result is what is known as the reproducibility crisis. And Nature magazine did a survey of 1,500 scientists, and the title of their article was 1,500 Scientists Lift the Lid on the Crisis Rocking Research.
I don't know which slide you want to be on.
Number 5, please. So 52% of the researchers said that, yes, there is a significant reproducibility crisis in preclinical research. I started this company a lot because of visiting a cell culture lab and seeing how it hadn't changed in 20, 40 years. But then I read a study in 2013. I started Thrive in 2014. Amgen spent 10 years and had up to 100 scientists trying to replicate or reproduce 53 landmark oncology studies, most of which they had paid a lot of money for licensing in. They could only replicate 9%, 6 of the 53. And there's been study after study. No study has shown that more than 50% are replicatable. Further studies have shown that about 50% of those preclinical errors are the results of problems with cell culture. So all of this basically slows down drug development and is why there's about 2% of all drug programs actually succeed. So what Thrive is doing is industrializing biology. And when I say that things haven't changed in 70 years, I really mean it. When we have slides, I'll show you a picture of a almost exactly 70 years ago lab. And it hasn't really changed since then. So the first cell line was HeLa cells at Tuskegee Institute. And things haven't changed. It's still a craft. There are still poor environmental controls, limited image and data capture, and it's not scalable. And do we have slides, Marty?
We're on slide 6.
Okay. Oh, okay. I didn't see they were there. Great. Great. Well, there's the picture from 1952, and it really is not done much differently. After reading that article from Amgen about the reproducibility crisis, I went and visited a cell culture lab, and it was deja vu all over again because it really, it really just had not changed. So what we're doing for the first time in cell biology is we are creating about 10 times the quantity of data that genomics produces. Our labs are, our customers are producing 5 or so terabytes a week. And what we're really providing is the right data, enough data for 3 things. Number 1, for breakthrough insights. For number 2, vastly improved processes. If you don't measure it, you can't control it. And if you're not recording things other than in lab notebooks, you're not going to be able to improve the processes. So this data, number 2, improves processes. And number 3, in order to have truly intelligent automation, you need this kind of data, and you need it for data mining as well as artificial intelligence and machine learning. There's no data sets out there in live cell biology, and our plan is to create those. So I can tell you how bad it is, but and how good Thrive is by rather a customer tell you. So a chairman of a department of biochemistry at a leading medical school in the US described the current status of optical microscopy, cell culture imaging, as current tools are failing us because they hurt cells, only give us 2D, single point in time images, and don't capture contextual or enough data. And after using our instrument, he said, Thrive will change the world. Thrive allows us to understand and see living, dynamic cells and tissues in 3D over time with remarkable images. We're just trying to follow the biology. Next slide. Actually, 2 slides, sorry. Next one. Thank you. So just a little bit of background on Thrive. So we have a family of products, 5 of them, 2 of which we have introduced. The third one we'll be introducing this year. And there's no FDA approval that's required. I have run several small and mid-sized pharmaceutical companies and diagnostic companies, and I don't miss it. We're able to move a lot more quickly as a result. We have 23 units that have been purchased or ordered. 70% of them, kind of to my surprise, are from industry, for-profit companies. Usually opinion leaders are are academics, but corporations and pharma, biotech are spending huge amounts on drug development programs and are aware of these problems. So this year, we expect 4.4 million in sales. We've booked about half of that, and it's just the beginning of the year. And in 5 years, expect to be $150 million in revenue. I've done that in 3 other companies, actually 2 of them faster than that. And we will have sold a cumulative 1,100 units by the time we get to the 5th year. Now, competition is very limited. There's always competition for something. There's and in our case, it's the microscope, which also hasn't changed that much in 70 years. But there's very limited innovation in this field because it really is neglected. One of our customers called it an island of manual processes in an ocean of automation. So we're innovators in the field, and so we've been able to file really a large amount of applications, patent applications, 86 in 39 families. We've raised $34 million to date. The majority of that comes from family offices, such as the Estee Lauder family, which is invested in 2 of my previous companies. Angel groups, we have 7 angel groups and industry executives like CEOs of Novartis, Johns Manville, Exact Sciences. So we're currently finishing up, probably next week, a $9 million Series B convertible note, which will convert into a Series B mid-year. And we've got about 8.5 million of 9 million. Would love to fill that up over the next week. We're also in the process of acquiring a company that's a leader in environmental controls for cell culture. And that company is one that they're a vendor. We see them in almost every lab. One of the most important things, as I described what goes on in cell culture lab, you might have noticed, is the fact that these cells are crossing multiple environments. They don't have to. And then we expect a Series B for $15 million mid this year, and it might be our last financing. Next slide, please.
So Tom, it's just really phenomenal how there's so much guesswork involved in the space, in the existing space, and how it kind of filtrates through everything else that's in the industry.
Yeah, it's Marty's point was it's amazing how much kind of errors and manual. It's many industries have a dirty little secret that you only know about if you're inside, and cell culture is definitely one of the worst things I've ever seen.
It would never be allowed in the auto industry or
No.
the airline industry.
That's right. I mean, and as I said,
No Six Sigma, it's 2 Sigma.
yeah, it's absolutely incredible. And I found a similar situation at Cytic, where we automated cytology labs, which read Pap smears. And as a result of my company, now 90% of all Pap smears are read by our instruments. Pap smears went from every year to every 3 years, and we changed the course of cervical cancer. And those labs were just like cell culture labs. So we're going to do the same thing in cell culture that we did at Cytic with cytology labs. This one's harder because they're live cells as opposed to stained cells. But this is really abominable. It's just it's incredible.
I mean, one of the things that Fred Nazen, who's another investor from our group, has said many times is that nobody, besides the fact that you can't get correct data on single drug testing, most people over 65 are taking 3, 5, 7 drugs, and there's no testing on how all these drugs interact with each other.
Right. Yeah, a lot of that can be modeled. So one of the Marty, one of the largest areas of growth. So what is cell culture really trying to achieve? I maybe should have mentioned it. It's to allow us to model things like what you just mentioned, Marty, which is drug interactions, without doing it in people. That's what cell culture is for. And the industry has evolved to organoids and tissues, which give you much more complex but better answers rather than just thin layers of cells. But that's really what we're trying to achieve is a patient model. And our cells are so distant from a patient at this point, I mean, with contamination and mislabeling. So our family of instruments, we have in the upper left, a benchtop instrument, and we've also married that with a 50-plate incubator. These are all networkable. They all use the same databases, collect huge amounts of data. Most of our focus now is on software modules. We have 10 engineers, 9.5 are software, and we're building modules to replace the way infectious disease research is done, which is frightening. Try and get a couple of minutes in on that. To do organoid imaging, which is, I said, is a very fast-growing area, machine learning toolkit, collaboration platform, so the scientists can actually share those same data and images. Next slide, please. So it's not to solve the problem of cell culture and cell imaging. It's not just a better microscope or a different microscope. It takes a different approach, and there are multiple things that have to be addressed. So number one is control the environment, which is going to improve results and increase efficiency because experiments don't have to be redone over and over again. And you need documentation. Everything should be documented. Right now, penetration of electronic lab notebooks in cell culture is the lowest of any life science type of laboratory. It's at about 3%. And you have to acquire great images so that you can see things that you couldn't see before because you're actually able to follow the biology. And you have to automate that imaging and the culturing the cells because it has to be done the same way every time. And humans are just not going to image the same way every time. And so with our instrument, you just put the plate in, it pulls it in, and it takes gigabytes of images and then returns the plate and does it the same way. And robotics is better at doing things often than people. We need to be saving this data and populate the database, which doesn't exist. And in the future, we plan to have, based on our access to our customers' images, which we get the largest live cell biology database that exists. And this data has to be accessible so that there are feedback loops, it's easy to use, and you have to be able to analyze it. And we attack all of those. Microscopes only attack one or two of those. Next slide, please. So imaging is key, and we provide enough data and the right data. We provide comparable images, and comparability is critically important because right now, the same person doing the same experiment twice will get very different results and much less across labs, across time. So a lot of the problem is that preclinical research is not comparable from institution to institution. And a lot of this is not even comparable within a lab, which is why many labs will do an experiment on average 3.8 times until they get a confirmation of another one of their experiments. It's obvious that those are often two flukes, two wrong answers, but you got the same wrong answer twice. So that's viewed as science, which it's not. So we take thousands of 5-megapixel images. An average scan of a cell culture plate takes about 30 gigabytes of images in 10 minutes. It's about 200 times more than any other instrument that's available. And as I mentioned, our customers are generating terabytes a week. They don't even know it. We make it so easy for them, which you have to do because cell biologists are not engineers generally, and they think differently. So most important, there are two things that are most important about our imaging. One is we take images with at least 100 focal planes, 2 to 50 microns apart. So we're collecting data on things like jaggedness of the cell and texture and curling. We can identify 80 different morphologies that then can be correlated with, say, the effect of a drug and things like that. And unlike other instruments, we've achieved the holy grail, which is we can follow a single cell over months or a colony so that we can actually see the impact of the biology or the impact of a virus on the cell or an impact of T cells. And because we have those stacks, those multiple focal planes, we're able to image organoids, lab organs on a chip. We can image suspension cells, which is what's used in immunology where the cells are floating. No one else is able to get sharp images of all those cells. Next slide, please.
And you can track those images over time?
We can track those images for years. So we have really for the first time traceability, and you actually know whether it's monoclonal, what the health of it is, and you know it's not mislabeled. So why would somebody need our instruments? Well, we fit into the current daily lab workflow. We provide access to the plates easily, even though we're imaging them. We image them for only a short period of time. And we use computers to more thoroughly inspect them. Computers do a much better job. At Cytic, we built a system. It was a machine vision system. Cytic was an AI company. And 90% of all pap smears are being read now using AI, not something that's talked about. So I've had, of my 7 unicorns, 3 were AI companies. Nobody knew that they were AI companies. By definition, they were, by the way other people, but it's a technique that, that we used. So our system automatically documents everything with barcoding. We make it very easy to record what reagents are used, who the operator is, where the plate was within the lab. When we build these databases, and we have a collaboration platform as part of our software, so researchers can, in fact, compare data. And this results in better quality, well, bottom line, experiments. Next slide, please. So I mentioned competition is basically digital microscopes, but none of them have the quantities of data that we do, the advanced focus algorithms that we've developed, the extensive analytical tools, the kind of database. And we've also developed, which is helps us with competition, incredible IP portfolio of 86 patent applications in 39 patent families, and that's across software and hardware. So next, oh good, you're already there. So here's some images that from our instrument. In the top middle, you'll see a graph with lots of curves in it. That's something that wasn't able to be done before our instrument. That's actually the effect of 50 different drugs that were screened on cells that they were studying wound healing, and were never able to do that kind of a screening before. And just to the right of it is a viral plaque assay, where for the first time, researchers did not have to stain their cells to study the effects on viruses because we could see it without staining. Well, what's the importance of that? When you stain, you kill the cells, and so you're not able to study the continued evolution of them. Next slide, please. I'll skip that one. The next couple. So I just want to talk about the size of the market briefly. This is a $29 billion market. There's 60,000 labs that are accessible. We're selling through a direct sales force. And the drivers for growth are that we're expanding into all areas of life sciences. Prior to COVID, next one, we didn't know that we were relevant to infectious disease, but it's actually our most prevalent application.
This one here?
No, next.
This one here.
Actually, I'm going to go to 18, the next one. Thanks. So just want to give you a snapshot of our customers. So we've delivered and have orders for 23 instruments. That's a value of about 3.8 million. This past quarter was our first commercial quarter. We booked 1.1 million. To my surprise, as I mentioned, 70% of our customers are from industry. I call it biology as a business, which is what they're doing. The applications were split very evenly across drug screening, stem cells, infectious disease, and a little bit to my surprise, process control. We're getting pulled into bioprocessing, which is even a bigger market than the research market, much quicker than we thought. Next slide. So my own background, I mentioned, I've started 17 companies. My first 3 were AI companies. AI has been a part of most of the companies as a technique. I have a really brilliant, and I've worked with many chief scientific officers and co-founders, a really brilliant one. Alan Blanchard's responsible for most of the sequencing instruments that are out there. The giant in this field is Illumina, and he developed those sequencers. Illumina actually acquired a company that had them. In this industry, innovation is bought, which is probably what will happen to us. Next slide, please. So just next one. So to summarize, we're transforming the field of cell-based experiments, which is fundamental to biomedical research. We're bringing to cell biology what sequencing brought to genomics, which is lots of reproducible data. And we're providing critically needed data to improve drug discovery. And ultimately, our vision is to speed drugs from lab to patient and with better than 2% chance. Again, I'd rather say about what customers say about us than what I say about us. Some of quotes from our customers are remarkable resolution. We see things we have never been able to see before. Tracking single cells and colonies in 3D is game-changing for drug discovery. This is what cell culture and cell imaging should look like. So thank you very much, Marty. I hope I left a little time for questions.
Are there any questions here at this point, or I mean, this really ties in exactly to what the panel before was talking about when it comes to data, and also, I mean, I really think that data, if you do like a Six Sigma approach, it's probably like 2 or 3 Sigma right now in the space. I mean,
Which is pitiful, right?
which is not tolerable in
not
any other
tolerable.
industry, right?
Right.
If you had highways that were 3 Sigma, everybody be crashing and sinking through holes,
Right.
right? So.
And we spend huge amounts, $50 billion a year is spent on preclinical research, and it's based on cell culture that's manually done. So I am around today and tomorrow and would love to talk to people, or my email is tom@thrivebio.com. And we're trying to finish up, I think we will, the $9 million convertible note next week. Would love participants in that, or in our acquisition, or in our Series B later in the year.
Will you be at dinner later on tonight?
Yes, I will.
And so he'll be at dinner also. There'll be other people that weren't here. So
Great. I don't want to miss any of your dinners.
fantastic. Great.
Thank
Well,
you.
listen, Tom, great job. Thanks so much.